Okay, everybody. So today we'll be discussing about the regulation of glycogen breakdown. So let's understand what exactly I'm talking about. I'm going to talk about glycogen breakdown regulation. So how is it regulated? The idea that I'm trying to discuss here with you is that glucose, you store up your glucose in the form of glycogen so that you can use it when the time comes. Now, the major storehouses of your glycogen, one is liver, the other one is muscles. Now, whenever you have a need, both of them are going to break down the glycogen, and they're going to use up use of the glucose which they've made from glycogen. How does it happen? I'm going to show you a beautiful slide here that will sort of wrap up the things for you. So the idea here again is that this is your liver and this is your muscle. So both of them are the storehouses of glucose, and the basic understanding is we want to break down this. We want to break down this glycogen into glucose 1 phosphate, because this glucose 1 phosphate is the one which we are going to use to drive energy. Now, the whole idea here is going to be based on this thing, which is the glycogen phosphorylase enzyme. Now, this glycogen phosphorylase enzyme, it can be active and it can be inactive. Here it is inactive, here it is active. When you add phosphate to it, that is when when it is phosphorylated, it is active. When you remove phosphate from it, it becomes dephosphorylated, it is inactive. Let's see what's happening. When the enzyme is active, it breaks down glycogen to glucose 1 phosphate. Okay, that should be clear to in your mind. Now, this is this enzyme called phosphorylase kinase, which will also be called as PK in short. This PK enzyme is the one which phosphorylates glycogen phosphorylase, meaning it's going to help in the breakdown of glycogen to glucose 1 phosphate. There's another enzyme which is called phosphoprotein phosphatase. Ase means it's going to remove phosphate, thereby deactivating this enzyme. So we need to focus on these things. I'll let you know what things you need to focus on. You need to focus on this final step, that is we have to break down glycogen to glucose. The enzyme responsible has to be activated, meaning phosphorylated glycogen phosphorylase, and this is brought about by PK. So what you need to understand, thereby what you need to understand is, if you want to break down glycogen, you want to activate PK. And what are the substances that activate? See, this PK is going to finally activate glycogen phosphorylase. So you want to activate PK. Now, what activates PK? Your liver has this. Now, your liver has glucagon receptors. Remember, this has glucagon receptors, but your muscle does not have glucagon receptors. So when you have glucagon in your body, in response to stress or anything, it goes and acts on your liver, thereby, sort of increasing camp in the liver, finally activating phosphorylase kinase and eventually making glucose out of glycogen. That won't happen in muscles, but muscles do have epinephrine receptors, and they also do the same shit. They increase the camp, thereby this cascade is stimulated. It started. But we must remember that in muscles, the more important stimulus is going to be the release of calcium from the sarcoplasmic reticulum. Whenever the muscle contracts, it releases calcium outside the sarcoplasmic reticulum, and that calcium is responsible for the breakdown of this, for the activation of this enzyme. So again, this cascade goes on working. So what do we understand? We understand that the glycogenolysis is regulated in this way. Muscle contracts, calcium comes out, activates phosphorylase kinase, that further activates glycogen phosphorylase, finally breaks down glycogen to glucose 1 phosphate. And that sort of wraps up your regulation of glycogen regulation. Thank you.